to everybody. Welcome everybody. Our first speaker is Mirko Martini, and this is the molecular evolution session. Mirko, we're good to start whenever you're ready.
Yes, thank you. Hi everyone, my name is Mirko Martini. I'm a PhD student at the University of Bologna, and today I'll present you the results of our latest research project on convergent mechanisms, genetic mechanisms underlying lifespan evolution in avian species. There are many different definitions of aging, but one of the most comprehensive one is the inevitable process consisting of the progressive deterioration of physiological function of any biological object, from a cell to an entire organism. Humans have always been fascinated by aging, and this fascination has grown in recent decades as the world population age and the gap between health and lifespan continues to widen. There are many different theories that want to describe aging, from mechanistic to evolutionary one. With our work, we want to study the existence of common convergent genetic background able to support longevity extension or shortening in birds. This particular vertebrate class is gaining more attention over the years due to its intrinsic characteristics, like great variability in their lifespan distribution, and the fact that birds can live 3 to 4 times same size mammals. So we, for 141 other species, collected genomic resources, their genomic resources, and 2 life history traits. In particular, we focused on the maximum lifespan and weight. The first difficulty that we encountered was how to define long-lived species. In particular, we went for the most straightforward way and split the maximum lifespan distribution into quartiles. We labeled long-lived those species having their maximum lifespan in the upper quartile, and we labeled short-lived species those that had their maximum lifespan in the first quartile. For the first time, we included short-lived species in a longevity study because we thought that we could delve deeper into aging regulation, studying both extremes of the lifespan distribution. This is the pipeline that we used. This is a multi-strategy pipeline that included convergent evolution analysis with Toucher, codon-based selection analysis with CodeML, and then inference of protein-protein interaction network using string with subsequent analysis using cytoscape and functional enrichment. This is the species tree that we used. With this representation, we could understand the distribution of our maximal lifespan within our phylogeny. In particular, we did not infer a new topology because the phylogeny of Avian is pretty tough to decipher. So we decided to use an already published topology from Kuhl et al. In 2021. The very core of the work was the convergent analysis that exploited the program Toucher. That Toucher is built on relative evolutionary rates that are here used as a proxy of the molecular evolution of amino acid sequences. First, Toucher calculates relative evolutionary rates for each species for each gene tree, standardizing this gene tree with the species tree. Then it compares rares between each pair of species having and not the trait of interest, and the results are then compared to a null distribution obtained from a permutation of the dataset itself. Results are lists of orthogroups that are more probably convergently evolved, and this convergence can be labeled in 2 ways: constrained if the if it is hypothesized a purifying section, and accelerated if it hypothesized a positive or relaxed selection. We decided to represent the results of our of our structure analysis as density distribution of the p-value of the inference itself. We can observe from both short and long-lived analysis that there is a peak in the most left part of the distribution that is linkable to an enrichment in a significant p-value. This peak is absent from the underneath gray distribution, that is the distribution obtained from 10 random controls performed on same size groups obtained from randomly chosen species within those that do not present the trait of interest. We can observe that the two analyses differ in the genes that have been identified as significant and as the proportion of constrained and accelerated genes. In particular, we obtained more constrained genes in the long-lived analysis and more accelerated genes in the short-lived analysis. We knew that the structure was prone to false positives. For this reason, we further filtered our results with CodeML in order to understand which gene not only presented a constrained or accelerated evolution in their amino acid sequence, but also showed signatures of purifying or positive or slash relaxed selection in the nucleotide sequence. So we used a branch model where we computed two classes of omega values, one for terminal branches of species having the trait of interest, and one for terminal branches of species not having the trait of interest. The results were plotted as the differences between the omega value of species having the trait and not. In particular, we confirmed that constrained genes indeed had a smaller omega value in the terminal branches of species having the trait in comparison to those that do not have the trait. And the same is true in an opposite way for accelerated genes with a greater omega value in species having the trait of interest. Those genes that were identified as significant after this filter were labeled as concordant and were used to identify and compute the network, the protein-protein interaction network. We obtained a network of 86 nodes that we split into 11 modules. Each module is here represented with a general term that represents the functional enrichment of the nodes and the protein present in each module. And clustering these representative terms, we can see that there are two groups of function already known for their pivotal role in the aging regulation. In particular, in the left part of the network, we can observe many different metabolisms, while in the right part of the network, we can observe different processes related to the cell proliferative process and cell regulation. Then we further analyzed this network when computing key describing metrics, and we, in particular, focused on stress and degree. Degree is the number of connections that are departing from a node, and here is represented as the dimension of the node, while stress is a value that wants to compute the importance of a node in connecting different parts of the network itself. And here is the width of the node's border. In particular, we wanted to analyze if there was if there were any particular pattern of these two metrics within our nodes. And indeed, we found significant results when comparing nodes grouped by their longevity label. In fact, genes identified in short-lived analysis had a significant greater stress than genes identified in long-lived analysis. And another great result was that genes already present, already known for their role in aging regulation, in particular in other organisms, for example, human, and for this reason, part of the human aging network, the human aging database, had a significantly greater stress and degree in respect to those genes that are not present in this particular database. So, in conclusion, we successfully found identified a convergent signal in a group of genes in short and long-lived animal, in long-lived birds. In particular, we can think that our analysis could support that there is a common genetic background explaining longevity in birds. While others only focused on accelerated evolution, we studied also conserving evolution, constrained evolution, because, and in fact, it was important because it resulted as the pattern most important in describing the evolution of genes in long-lived animals. Our multi-strategy pipeline successfully filtered genes that in the end, build a network with more connection than expected. And the inclusion of short-lived species repaid us because not only genes identified in short-lived species were part of the network, but having those genes at greater stress, they were important and essential in the network structure as we can observe it. In the end, the cross-reference done on the human aging genomic resource was important because it suggested the importance of those genes not present in the database, but sharing high value of metrics with those present in the database, proposing them to be the next in line for functional studies and deeper analysis. So for the real conclusion, I want to thank all my collaborators, supervisor, and my tutors for helping me and accompanying me in my PhD journey. And I want to thank you for your attention.
Perfect timing. Thank you, Mirko. Do we have any questions from the audience? You can raise your hand or write in the Q&A. I don't see anybody raising their hand.
No.
Okay. No questions from the audience. Mirko, I do have a brief question.
Yeah.
Have you tried other tools for the selection analysis, like the HiFi package, like Absroll for episodic diversifying selection, or MEME? Because I wonder how different the signals will be with different approaches. Not that it's not, it makes perfect sense, but I'm just curious, really.
Yeah, we actually tried other tools, and we actually started using those tools from the like MEME. We tried to use Relax and Absroll. Yeah. But actually, we preferred a more plain approach because we actually use CodeML as a filter for our significant genes from structure. So we could finely tune CodeML in order to be able to detect the hypotheses that we had. So able to see in the program itself and be able to, for example, extrapolate the omega value that instead it is quite difficult to do with Relax because Relax and other tools alike, they are like sometimes a black box. So they give you a good result and an important result that can have a significance in it. But sometimes we don't know what happened during the analysis. And we wanted the raw data to plot, for example, this particular distribution in this way.
That's great. Thank you very much, Mirko.
To you.
Any other questions?
Q&A.
Hi.
There's also a question in the Q&A.
Oh, great.
Yeah.
So for CodeML analysis, what were the CodeML analysis for the 3-rate model, or did you consider long-lived and short-lived them separately?
We considered short and long-lived separate. Yes, actually, there was a part that I did not present because it was not the core of the analysis itself, but a group of genes were found with the same gene in opposite lifespan pattern, but with opposite evolutionary force. So we found actually some genes that were, for example, constrained in long-lived and accelerated in short-lived. For at the beginning of the analysis, we wanted to put our focus on this particular genes, but we did not develop these genes.
Sorry to interrupt you. We have to move on to the next talk.
Yeah.
No, actually, sorry, break session. We
Okay.
can continue. Sorry, I interrupted.
No,
My God.
no, thank you. No, don't worry. So actually, we split the two different analyses, and we refined. For example, I don't think that I can share with this particular representation, but there are 3 genes that are this one, the one here and here, that are, for example, genes that are in opposite life trait under opposite forces. So we tried to understand what this could mean, and we thought that they could be particularly interesting because they were suggesting the same gene in the same pattern with different evolutionary pattern can describe completely different and opposite phenotypes, actually. I hope to have made myself clear. Sorry, you're muted if you are speaking.
Yes, I don't see any other questions, but Mirko is here with us. So if anybody wants to ask something,
yeah, for sure.
Our Q&A. If you want to move to the next session, we can also do that. Oh, thank you, Mirko, very much. I'm sorry for trying to stop you.
Or
I think we can wait a few more seconds just to
yeah, sure.
make sure that everybody comes, since other talks might be ending up now. So people might be joining for your talk. Excellent, we can start. So our next speaker is Fernando Torres. So Fernando, the stage is yours.
Right. Thank you everyone for coming to my talk today. So I am going to talk about patterns of deep coalescence across amniotes and what they can tell us about the relationship between genetic and phenotypic variation. The study of which has been central to biological research ever since the rediscovery of the work of Mendel in 1900. And while today molecular data dominates biology, the use of these data sets to study mechanisms underlying the evolution of phenotypic systems is still an underdeveloped area of research. And so in our study, we are aiming to contribute to our understanding of this link between genes and morphology by using a classic scientific approach that finds its origins in Hegelian dialectics. And that is, instead of holding conflicting ideas as opposite to each other, we investigate the sources of these conflicts to develop a new understanding. And so the first source of conflict we're looking at is that between genes and morphology, for which we turn to the classic example of the phylogenetic position of turtles, where molecular and morphological data have historically produced competing hypotheses. And while most data supports turtles as sister group to archosaurs, there is well-supported morphological hypotheses that are probably telling us something about the biological reality of these organisms. And so we're attempting to better understand mechanisms underlying morphological evolution by looking at the sources of these conflicts. The other type of conflict we're mainly looking at is that between gene trees and species trees. And so while we would expect that most genes are following speciation patterns, this is not always the case. Sometimes deep coalescent genes can produce a pattern that does not match the pattern of speciation events. this being the result of retained polymorphism throughout a divergent lineage. And this pattern of discordance between gene trees and species trees as a result of polymorphism is what we call incomplete lineage sorting, or ILS. And so with all this in mind, there are two main questions guiding our research. First, what are the patterns of gene trees to species trees conflicts across amniotes? And do we find higher levels of incomplete lineage sorting around clades with a history of morphology and gene conflicts? So to study this, we started by acquiring whole genome sequence data for 23 amniote species. We then performed ortholog selection using OrthoFinder and a custom Python script, which resulted in 1,074 orthologous genes across all these species. We then proceeded with standard sequence alignment and phylogenetic inference of gene trees and species trees. We then used this data to test our ILS hypothesis using a relative frequency analysis that shows the topology support of gene trees around 3 focal branches of an inferred species tree. And to target major divergences across amniotes, we assess the topological support of these branches relative to their potential alternative topologies, for which we had to assign our species to one of these clades in our species tree. And so under ILS, the expectation is that most of gene trees will be supporting the species tree with some amount of gene trees supporting discordant topologies, and that deep coalescence and consequently polymorphism would be present not only in the discordant gene trees, but also in some amount of the gene trees that are supporting the species tree as well. So we found that 4 of our three focal branch groups presented less than 5% discordance, exemplified here with the clade of Archosaurus plus Avis, where, as you can see, discordance is minimal. On the other hand, the remaining groups, 5 of them, be precise, presented more than 20% discordance, where some groups, like the Avis plus Neognathis, presented moderate levels of discordance, and we found that the highest levels of discordance were present in the reptilian group, here presented here, with about 32% of gene trees supporting discordant topologies. And so to further test our ILS hypothesis, we performed divergence time estimations for the external and the internal nodes of the groups of interest around all our gene trees, with the expectation that under ILS, we would find that the estimated dates coming from genes that are in conflict with the species tree will present older dates than those that come from genes that support the species tree. This we expect to find for both the external and the internal node, given that by definition, ILS has to go through multiple speciation events. And so when summarizing the data, we would expect to see something like this, where on average, the estimated ages coming from genes that are in conflict with species tree will present older dates, as I mentioned before, compared to the ones that are supporting the species tree. And this is what we find throughout most of our data. For example, on the Avian clade here, the gene trees that present the older dates are in fact those that are discordant with the species tree, supporting ILS as the source of discordance, this being true for this internal node and the external node as well. But this was not the case for all our groups of interest. In 2 cases, in reptilian and the mammalian groups, we find a slightly different pattern, where in the internal node, the genes that are presenting older dates are the ones that are discordant with the species tree, supporting again ILS as the source of discordance. However, for the external node, the pattern is reversed, and we find that the genes that are concordant with the species tree present slightly older dates. how can we explain that we have this finding that despite all the other data is supporting ILS as source of discordance, we find in these groups that the genes that are concordant with the species tree are presenting earlier dates that we were expecting. Well, this might be explained by an unconventional deep coalescent scenario. Under a regular deep coalescent scenario, we would have that on top of ILS genes, most of genes supporting the species tree will not be deep coalescent, with some amount of these genes supporting the species tree being deep coalescent. And this is going to result in the pattern that we see throughout most of our data. However, if we think of a scenario where on top of the ILS genes, only a few genes supporting the species tree are not deep coalescent, and actually the majority of these concordant genes are deep coalescent, but this is going to result in the pattern that we're seeing across reptiles and mammals. And this means that there is a larger amount of polymorphic genes across the stem of these clades in a minimum polymorphic interval that is marked not only by discordant ILS genes, but also by a great majority of the concordant genes as well. And what does this tell us about the morphology? Well, if we can think of a taxon that diverged during one of these polymorphic intervals, we can see how just by chance this taxon could have inherited either of the forms of this polymorphic gene. And now if we start thinking of this more as a species tree, and we consider a gene that is heavily contributing to the development or of a phylogenetically informative phenotype, of which there's many examples of, we can see how the resulting pattern can be interpreted as a convergence or a reversal of states, but it's actually representing the expression of shared ancestral polymorphism. And now, if we keep sampling an area of the tree within this polymorphic interval, then we would find a widespread homoplasy across multiple phenotypic systems. And this is, interestingly, exactly what we see near the origins of reptiles, mammals, and many other animal groups that we didn't sample in our study. But these findings highlight the importance of better understanding this type of dynamics, particularly given that we rely on the fossil record to do things like ancestral state reconstructions and for calibration points to date divergences. Well, to summarize, we found that ILS is widespread across amniotes. This was supported by our divergence time estimations across gene trees. We found that there is some clades, particularly reptiles and mammals, where we find more polymorphism that we were expecting, and that the discordance between morphology and molecular data might actually represent different samplings of a complex evolutionary dynamic. Moving forward, I think it'll be important to start examining these patterns across the tree of life, and if found to be widespread, as our data suggests, we should start accounting for these patterns in our models for phylogenetic reconstruction using morphological data. And finally, that we would need to start investigating more closely the genetic and developmental mechanisms underlying phylogenetically informative traits. Finally, I want to thank my two co-authors, Gabriel Beaver, who's my advisor, and longtime collaborator, EJ Juan, who's got another presentation going on later today at 4:30. Encourage everyone to go see that on the Ernest Mayer session, particularly anyone interested in phylogenetic methods. Also want to thank everyone at the Center for Functional Anatomy and Evolution, especially everyone at the Beaver Lab. I'm ready if anyone has any questions. Thank you.
Great talk, Fernando. Thank you. We do have a question in the Q&A. The question reads, "Hi, Fernando. As far as I know, the OrthoFinder will output the gene tree as well. Why still perform extra analysis to generate the trees?"
So the gene trees that come from OrthoFinder, first of all, not all the orthologous groups we grabbed as they come from the output from OrthoFinder. That resulted in, I think it was about 10 single copy ortho groups, which was the ones that we could have used for this. And that's why we had to use our custom Python script that looked for other ortho groups. And well, to keep it short, it'll just find ones where to extract only the ones that will show orthologous genes for all the taxa involved, to kind of remove any kind of paralogous ortho groups. The total number of ortho groups was something in the tens of thousands, but that was still reduced to our sample of 1,074 genes. So, well, in short, we didn't grab the output directly from there. We had to do an extra layer of filtering to be able to get the data that we got.
Okay, great. There is another question, but we are at 14 minutes now. So if I'm gonna release the question in the Q&A so you can answer in the chat if that's okay, and we can
Yeah,
go on to the next speaker. Sorry, that's the timer. So the next speaker is Savanna. Savanna, let's wait a few more seconds and you're good to start. Okay, I think we are good to go. So I'm muting myself. Savanna, the stage is yours.
Awesome. So I will jump right into it. Today I will be discussing my current PhD work on transcription factor motif evolution with gains and losses of social behavior. So as we all in this space know quite well, a single genotype can produce highly variable phenotypes. So for example, differences in gene regulation can produce different sexes, as seen in temperature-dependent sex determination in sea turtles. Differences in gene regulation can also produce different morphologies, as seen in species of butterflies that have different wing coloration based on the season they develop in. And differences in gene regulation can also produce different behaviors, as seen in species of locusts that exhibit solitary versus gregarious feeding behaviors. So this prevalence, where gene regulation is facilitating the emergence of different phenotypes, is seen across many taxa. And therefore, the question, how does gene regulation shape the emergence of phenotypic variation, is an important and prevalent question throughout evolutionary biology. And bees are an ideal model for investigating phenotypic plasticity, investigating this phenotypic variation, since they have experienced multiple independent evolutionary transitions between solitary and social life histories, and this enables comparative analyses of these social transitions across these lineages. So previous literature has correlated the expansion of predicted transcription factor motifs with sociality, suggesting a generalized mechanism involved in social evolution. So what I really mean is that the pattern that has been identified is that there are more predicted motif sequences for transcription factors to bind to in the same gene in the promoter of a social species compared to that of a solitary species. Like we see here in this cartoon, right? It's the same gene, but in the promoter region delineated in red for the social species, there's increased predicted motif binding compared to that of the blue for the solitary species. So this pattern suggests that this gene regulatory expansion is a generalized mechanism involved in social evolution. And with previous literature suggesting this generalized mechanism of greater transcription factor binding in social lineages, I further have inquired, are convergent transitions to and from sociality driven by shared changes in gene regulation? And furthermore, does regulatory complexity decrease in lineages that has secondarily lost that social behavior? So in order to address and answer these questions, I've developed a pipeline based on previous literature, which is a little complicated, and I would love to discuss in further detail with anyone via email or after this presentation. But for now, I'll highlight some of the important steps that I've taken in my methodology. So I first used 42 different publicly available bee genomes representing a range of social life histories. So we categorized species as ancestrally solitary, and those species were ones with no history of the lineage expressing any social behavior. We classified and categorized species as social, and these were all species with a reproductive division of labor, so they must exhibit that non-reproductive worker caste. They have cooperative brood care and overlapping generations in one nest. And finally, we categorized the life history strategy of secondarily solitary species, and these were species for which the ancestral state was social, but the lineage has now secondarily reverted to a solitary life history strategy. So after acquiring these genomes, I then used peptide sequences to identify orthogroups shared across these 42 species using OrthoFinder, which we've heard a little bit about today. And essentially, this step identifies the shared sets of genes, which allows for comparative analyses across these lineages. And for OrthoFinder, for my study, it identified around 13,000 orthogroups across my 42 species. So OrthoFinder also generates a phylogenetic tree, and that's the first result that I'd like to show you. And this is the phylogeny that OrthoFinder generated, and it supports the independent gains and losses of social behavior like we've talked about earlier, and the general relationships among the species seen here agree with previously published phylogenies, so we're good to use it. And the lineages I've included represent 3 independent gains of sociality, indicated by those red triangles, as well as 6 recent and independent losses of sociality, indicated by those yellow X's. So including these independent gains and losses of sociality, it enables this comparative approach to study changes in motif sequences with these social transitions. I then used PWM scan to scan promoter regions of these orthologous genes for occurrences of predicted transcription factor motif binding, and we sourced those motifs from Jasper's insect motif database. Finally, I ran 2 phylogenetic generalized least squares analyses that analyzed the correlation between sociality and motif occurrence. The first PGLS was for social gains, so from that transition from an ancestrally solitary to a social state. And secondly, we ran it for social losses, which is that transition from a social state to a secondarily solitary state. And I'll now present the results from running these analyses. So results from our first PGLS, looking only at gains in sociality, which is the transition from an ancestrally solitary state to a social state. So each bar in this graph will represent one of the motifs tested and their associated orthogroups across the species. So on the top half of this graph, in red, in the red part of that bar, is the number of social biased orthogroups associated with that specific motif. And then the bottom half in blue of that bar represents the number of solitary biased orthogroups associated with that motif. So when examining the number of social and solitary bias correlations for each motif, so what's that difference between that red portion and that blue portion, that social bias and solitary bias for each motif, we see that 213, so roughly 75% of the ones tested, were socially biased, meaning more orthogroups had higher motif counts for social species compared with ancestrally solitary species. These results suggest that, similar to previous findings, transitions from solitary to social behavior in bees are associated with an overall expansion in predicted transcription factor binding sites. So now, looking at the results from just the second PGLS, so looking at only losses in sociality, the transition from a social state to a secondarily solitary state, again, the bars in this graph will represent one of the motifs tested and their associated orthogroups across all species. And so on the top half of this graph in red, the red portion of that bar represents the number of social biased orthogroups associated with that specific motif. And on the bottom half, the yellow portion represents the number of solitary biased orthogroups associated with that motif. And this is for losses. So we first see that the overall number of orthogroups associated with each motif is significantly less than that of what we just previously saw in the gains, right? These bars are much smaller. And when examining the number of social and solitary bias correlations for each motif, so again, what's that difference between that red portion and yellow portion of that bar on the graph? We see that 205, so roughly 72% of those tested, showed an overall solitary biased correlation. So meaning more orthogroups had higher motif counts for secondarily solitary species compared to that of our social species. So we did not observe a widespread contraction of predicted motif presence in these secondarily solitary lineages, suggesting that many transcription factor motifs are in fact more prevalent in these secondary solitary lineages compared to that of the social. So since our motif level analyses of losses in sociality revealed these unexpected expansions, we decided to analyze promoters that were significant in both PGLSs to ask specifically whether promoter regions that were being implicated in these predicted motif expansions, so those that were significant in the first PGLS, were they also continuing to experience these expansions with losses, so in that second PGLS. We identified 149 motif OG pairs that were significantly correlated with both gains and losses, so significant in both PGLS models. These pairs, motif orthogroup pairs, represent the subset of promoters that exhibit significant regulatory changes from both the ancestrally solitary to social life history strategies and from ancestrally social to secondarily solitary life history strategies. So if motif changes are simply expanding randomly with time, then we should see the majority of these pairs being socially biased with gains and solitary bias with losses, so located in this bottom right portion of the table. However, we find a striking pattern in our promoter-specific analysis where 146 of the 149 motif OG pairs are either socially biased for both gains and losses, so located in that upper right-hand corner, or solitary bias for both gains and losses, located in that bottom left corner, indicating complementary changes with gains and losses of social behavior in this predicted transcription factor motif binding. We took this one step further and assessed whether these 149 significant motif OG pairs exhibit consistent patterns across the 42 species tested in our PGLS. And we did this through a visualization of their normalized motif counts in a heat map. So within this complicated heat map, which I'll bring up in a second, each column represents a species that was tested, those 42 species, and each row represents one of those significant motif orthologous pairs. So unsupervised hierarchical clustering of these motif OG pairs found significant in both transitions to and away from sociality revealed a clear structure where socially and solitary biased motif OG pairs are naturally grouped into distinct row clusters. So we can see on the left-hand side that the group of red rows grouping together, which are those motif OG pairs socially biased for both gains and losses, and we can also see the group of green rows clustering and grouping together, which are those motif OG pairs solitarily biased for both gains and losses. So this unsupervised hierarchical clustering not only is grouping by these convergent transitions, but it's also clustering as a result of social behavior. So across the bottom, they're tending to group together the red shortened species names, which are social, and for the most part, the blue and yellow for ancestrally solitary and solitary species that have recently lost sociality are grouping together in yellow. So this emergent structure from unsupervised hierarchical clustering is highlighting the strength of the phylogenetic signal and regulatory convergence in the motif regulatory changes that we're seeing to be associated with sociality. I can get there. So what I really want you guys to take away from this, and in conclusion, is that we're seeing evolutionary gains of social behavior being associated with widespread TF motif expansions. Losses of behavior we are seeing to not be associated with widespread transcription factor motif contractions, which could be due to a lack of evolutionary time. Losses only stem in our tree from around 20 million years ago. And finally, we see promoter regions of orthologous genes are convergently modulated in association with social evolution. So we see our promoter regions being socially biased for either both transitions to and from sociality or solitary bias for both transitions to and from sociality. So these results are suggesting that not only are these expansions associated with the evolution of social behavior, but specific regulatory targets may be convergently modulated across these gains and losses. So I'd like to thank my lab at the University of Kentucky. And I don't know if we have time for many questions, but please shoot me an email. I would love to chat further.
Great talk, Savanna. We've just reached 14 minutes since you've started. We might have like a minute if people want to ask questions or write them in the Q&A. And yes, there is a question. Zamfa, can you unmute yourself?
Yes, I got this in me. Yeah, very interesting talk, Savanna. I was just wondering, even with 146 motif changes significantly associated with sociality or loss of it, that's still a huge number. I was just wondering if all of them are associated with, or some of them could be associated with some other things that correlate with the change in sociality. So there may be some other developmental changes that associate or correlated with the change of sociality. So could be just a small number of those are related to the change of sociality. And also related question is, do you have any idea how the motif changes and at a molecular level, at a mechanistic level, how that relate to the change in sociality?
Yeah, so like we
Sorry
have
to
mentioned,
interrupt you all. I think we need to move to the next talk, but if Savanna, you
I will
would like
message.
to... Yes, sorry for